Welcome to the recap on enzymes with the logical llama. So our main question here is, what exactly are enzymes? Well, we learned in macromolecules that enzymes are proteins. So to add to that, these are specialized proteins that are going to serve as biological catalysts for many of the reactions that take place in our bodies. Enzymes are working as biological catalysts. And so how exactly does that happen? They're going to work by speeding up chemical reactions by actually lowering the activation energy. And the activation energy is the amount of energy that is needed to start a reaction. So we're going to have a graph, and the graph is going to have two lines, a purple line and a green line. The purple line is representing the rate of a reaction that does not have an enzyme assisting it, and the green line does have an enzyme assisting it. The height or the peak of the line graph represents how much energy is needed to start the reaction, both ending at the same point. The enzymes allow for the reaction to happen faster or quicker. Something that is extremely unique about an enzyme is that they are very, very specific. An enzyme and a substrate have to fit perfectly together in order for the enzyme to function. If they do not fit like two puzzle pieces, the enzyme will not work. So each enzyme has its very own substrate that will work without any other substrate. This is referred to as the lock and key model, where the enzyme and the substrate have to match like puzzle pieces perfectly. So there can come a point where an enzyme no longer functions, and this is referred to as denaturation, or to have the enzyme denature. All enzymes will have an optimal or an ideal temperature and or pH. And if we take an enzyme outside of their particular optimal range, the enzyme is said to denature. So whether we make it too hot, too cold, too acidic, too basic, the active site and substrate will no longer fit together. Denaturing causes the enzyme to actually unfold, which will change the active site shape. Therefore, the enzyme no longer functions properly. Once an enzyme has been denatured, it will never function again. Before we spoke about the lock and key model, the enzyme substrate complex model is basically the same thing, but more sciencey. So you see a diagram with the enzyme substrate and then how the enzyme turns that substrate into products, okay? The process of breaking it down. So there's a place where the chemical reaction actually occurs, and that is going to be where the substrate and enzyme fit together perfectly. When they are matched up like puzzle pieces, that is when the substrate gets broken down into the actual products. So what's the big deal with enzymes? Why are they so important, and why are we learning about them? Well, nearly every single reaction that occurs in our bodies requires help from some enzyme. And if our bodies did not have those particular enzymes necessary to sustain life, those reactions would not occur fast enough, and ultimately we would die. So thank you, enzymes. Well, that's it for our recap on enzymes. This is the Logical Llama saying see you next time.